Today we are talking about a brand new paper. It just came out, and it's an absolute game changer for large-scale genome editing. And the secret? Well, it's all about teaching our therapeutic tools how to be invisible, how to fly right under the radar of our body's own defense systems. Think about it. What if we could do more than just fix a single letter of DNA? What if we could go in and swap out an entire faulty gene, thousands of base pairs long, with a brand new, perfectly working copy? That's been the holy grail of genetic medicine forever. But there's always been one massive roadblock standing in the way. And that roadblock? Well, it's us. It's our own immune system. The very security system that's designed to protect us sees these amazing gene therapy tools as a dangerous invader, and it launches an all-out attack. It makes the cure itself toxic, and sometimes just plain impossible.
All right, so here's how we're going to break down this incredible scientific quest. First, we'll look at the roadblock that had scientists stumped. Then, we'll see the genius DNA in disguise they created to sneak past it. We'll get into the nuts and bolts of their clever install method, check out its success in the real world, and then we'll dream a little about what this means for the future.
So, let's dig into this first part. Why does our body put up such a fight against something that's supposed to be helping it? It seems kinda counterintuitive, right?
So, to do these massive edits, scientists use these amazing enzymes called recombinases. You can think of them like molecular surgeons, or maybe locksmiths, that can cut and paste enormous chunks of DNA with unbelievable precision. They are the perfect tool for this kind of job. So the problem isn't the surgeon itself. The problem is the package it's carrying, the DNA template. Historically, that's been a long piece of double-stranded DNA, or dsDNA for short. And to your cell's security system, that looks exactly like a virus that's trying to invade.
And the second that dsDNA gets inside the cell, all hell breaks loose. It trips these powerful alarms, like the cGAS-STING pathway, that unleash a full-blown inflammatory storm. This causes massive cell death, it's incredibly toxic, and it makes the therapy itself dangerous. This right here is why this amazing technology has been stuck in the lab for so long.
So how in the world did this team of scientists figure out a way around this? How did they create the perfect disguise? Well, this is where it gets really brilliant. They took a page right out of the enemy's playbook. They learned from viruses. And the solution is it's so elegant. Instead of that big, clunky, linear, double-stranded DNA, they created a circular, single-stranded DNA molecule, or cssDNA. You can think of it like a stealth agent. It's designed to look nothing like the intruder the cell is trained to hunt down.
And when you put these two side by side, the difference is just night and day. The old dsDNA screams danger to the cell's immune sensors. But this new cssDNA, it just slips right past them. It's way less toxic, and it's just fundamentally a much, much safer way to deliver a gene.
Okay, so having a stealthy package is amazing, but that's only half the battle. Your molecular surgeon, that recombinase, still has to be able to actually use the DNA inside. And that's where the team developed this really clever method they call install. And here is the final piece of the puzzle. These recombinase enzymes, they evolved over millions of years to grab onto double-stranded DNA. That's the only thing they know how to work with. So how do you get them to recognize this new, weird, single-stranded, circular version? It's a real head-scratcher.
So their first shot at it, install 1, was kind of passive. The idea was you get the cssDNA into the cell's nucleus, and then you just hope that the cell's own machinery eventually builds the second strand for you. It does work, but it's really slow and not very efficient.
But this is the real magic. Install 2. What they did was pre-attach a tiny little piece of DNA, called a pip, right onto their stealthy circle. And what this does is create a tiny double-stranded handle. It's just enough for the recombinase to get a grip, and boom, it can get to work immediately without waiting on the cell at all. It's genius.
Okay, so the theory is fantastic. You've got a stealthy DNA package with its own custom handle for the molecular surgeon. But does it actually work in a living system? Let's take a look at the results.
Okay, so first up, they tested this in primary human T cells. We're talking real, frontline immune cells. And the results are just striking. The new install method was over 4 times more efficient than the old way. That is a huge leap in performance. But honestly, efficiency isn't even the most important story here. Safety is.
And now for the moment that just punches you in the gut and shows you exactly why this research matters. When they used the old dsDNA method in mice, at a dose that should be therapeutic, the toxic immune reaction was so bad
that the fatality rate was 100%. The cure was literally lethal.
But then they tried the new install method, and every single mouse survived. And they weren't just alive, they were completely healthy, acting normally, even when they were given higher doses. This isn't just an improvement, this is a complete game changer for safety.
And here's the visual proof that it didn't just keep them safe. It actually worked. What you're looking at is data from the livers of those mice that survived. That clear band confirms it. The new gene was successfully installed right into their genome. It was both safe and effective.
So, what does this all mean? Let's zoom out and look at the big picture, because this could change everything.
You know, the researchers themselves put it perfectly. They said that install overcomes this key challenge by, quote, evading the toxic immune responses. They basically solved the central problem that has been holding this entire field back for years.
So, the huge takeaway here is that we now have a way to safely deliver gene-sized chunks of DNA without using viruses. This is a big deal because it lets us sidestep all the problems with current viral methods like AAVs, things like pre-existing immunity or limits on the size of the gene you can deliver, or the fact that you often can't redose a patient. This really does open the door to a whole new class of medicines.
And that leaves us with this incredibly hopeful question. For the very first time, it looks like we have a safe and effective way to write entire genes into living animals. So the question is no longer, can we do it? The real question now is, where do we start?